Hello and welcome to Online Lecture 6, or Great Experiments in Molecular Biology, also known as How Do We Know What We Know? I'm excited to talk to you about this lecture today. It has a lot of really interesting experiments and history and background information about the scientists behind some of the most important facts that we know about cells and biological molecules. So please be sure to watch this in its entirety and take notes, and this will help you get ready for our class on October 23rd. The topics in this lecture cover core concepts 2, 5, and 6, and core competencies 1, 2, and 3 that are listed on your syllabus. As far as the core concepts go, this lecture should be a review of 2, 5, and 6, but we're going to look at those topics in a new way, really emphasizing the core competencies listed on this slide: data interpretation, testing hypotheses and supporting scientific arguments, and problem solving and critical thinking skills. And we get to do this with a really interesting historical backdrop of these scientists as people and the world that they lived in at the time that they were doing these seminal experiments. At any point in time, if you click on this link in the title slide here, you can go to a very interesting website. And this is a really great website to get a feel for how the field of molecular biology developed. And one of my favorite things about it is that in addition to seeing seminal experiments and the order in which they occurred, if you click on the timeline, you can also see what was happening sort of socially at the same time. So I'm going to click here on the pre-1920s. And when you do that, you'll see some of the individuals I'm going to talk about in a little bit. Gregor Mendel, who we know worked on pea plants. A new gentleman who you'll be introduced to, Friedrich Miescher. Fritz was his nickname. And if you'll notice at the time they were doing their work, so right around the time Miescher discovered DNA, the telephone was patented, for example. So if you follow along, and here you click along and you get up into the 1920s, when Barbara McClintock was doing some of her work, you can see that this was just after women got the vote in the US and movies were actually invented, the talkies. I encourage you to explore this website. You'll find if you click on these individuals, you can see a biography. Here is Barbara McClintock's biography. And in some cases, you can also find their little animations or videos that you can watch as well. So if you want to know more, this is a fabulous place to go and find out more. Science as a discipline is really different from other types of inquiry. We talked about this at the beginning of the semester when we talked about the process of science. But we want to circle back to that now and really look at some concrete examples of how it is that science is different from those other disciplines. And the primary distinction being that we know the things that we know in science because we have evidence to support our claims. And typically this evidence is quantitative or experimental in nature. We're going to explore this idea using molecular biology as a backdrop because our current unit that we're focusing on, and really the rest of the class for the semester, focuses on molecular biology. Let's start with a familiar example, that of Gregor Mendel. You may recall this is the Austrian monk who did the work on the pea plants, which formed the basics for our modern theory of genetics. And as you know, Mendel worked on garden peas. He worked on them because they were easy to grow and cross, they grow very quickly, and also because they're tasty. I mean, let's face it, the monks were having to raise their own food and often provide food to the surrounding community, so he couldn't waste his time growing plants that weren't very good to eat. Now, the conclusions that Mendel came to, which you're already familiar with, were many, but one of these was that there were discrete units of inheritance and that he was able to predict their behavior in the gametes. Well, how was it that Mendel was able to do this? Let's take a look at the paper where Mendel described his work. Now, Mendel actually wrote in German, so we're very fortunate to be alive now because some really nice folks have actually taken Mendel's work, translated them to English, and posted them on the web. So I'm going to click on the link here, this Experiments in Plant Hybridization. And here we go. And now we can look at what Mendel did in his own work. The first thing that he has are some introductory remarks, and one of the things I thought was very interesting that Mendel noted is that Other people had worked on this task before him, and I like what he said. He notes here that these people have devoted a part of their lives with inexhaustible perseverance. And what a nice nod to give to his colleague. And in addition, he also acknowledges that that it's not surprising that no generally applicable law governing the formation and development of hybrids has been successfully formulated and can hardly be wondered at by anyone who's acquainted with the extent of the task and can appreciate the difficulties with which experiments of this class have to contend. So Mendel was saying, hey, these people have worked really hard, but we don't know, but we don't have a what's the phrase they use? A generally applicable law, because you know what? It's hard to do this work. And then at the very end, I find it very interesting what he says here is that his paper presents the records of a detailed experiment, and his experiment was practically confined to a small group. And here it is right here. And now after 8 years' pursuit, concluded in all of his essentials. So if you didn't read any further in Mendel's papers, one of the things I hope that you see right away is that to do science well often takes a long time. 8 years is much longer than a typical PhD, for example. So Mendel spent a lot of time out in the garden growing peas and very carefully doing these crosses and test crosses. He goes on to describe how he selected his plants, and I do encourage you to come to this link and read it in more detail if you're interested in this. For right now, I'm just going to scroll down a little bit further and show you the characters that he selected, and he's talking about things that we're already familiar with, such as differences in the color of the seed coat, the difference in the texture of the seed coat, and so on. He details the crosses that he does right here. And then very interesting, if you get a little bit further down, he comes and he actually talks about the results from his first generation of hybrids. So this is here in number 5. And here's experiment number 1, where he looked at the form of the seed. And this is what we looked at in class when we looked at the wrinkled seed coats versus the smooth seed coats. And he shows that he had 253 hybrids from which he obtained 732 seeds. And then he counted them and sorted them according to whether they had wrinkled seed coats or these round or smooth seed coats. And he found that 5,474 were round, whereas 1,850 had the wrinkled seed coat. And he did the same thing for the color, and then you'll notice therefrom the ratio 2.91 to 6 is deduced. And he does the same thing for the color of albumin, and he finds a ratio of 3 to 1. So in order to understand these results, you need to think about Mendel's first experiment. And remember what he did first was he took true breeding plants, so plants that always produced spherical seeds or plants that always produced wrinkled seeds, and crossed them back to themselves to make sure that those plants only ever produced a certain kind of seed coat. So those were the plants he started with, true breeding plants. And in his first cross, he basically did a cross between the two homozygotes. So that he started with, for this experiment that you're looking at right now, this was a cross between heterozygotes. And what he discovered is, that's funny, what he discovered is that when he crossed the heterozygotes, the recessive trait reappeared, and that the phenotype gave this ratio of 3 to 1, which he's showing here in the middle of the screen. And this is how this is the experimental basis for the idea of dominance and recessiveness, but also the idea that inheritance does not occur through blending, which was the prevailing theory of the time. Mendel did his work in an Austrian monastery, and it's interesting to think about what was happening at the world at the time that Mendel was actually doing his crosses. Remember, he did about 10,000 different crosses, so he was very busy working on pea plants, and his work was actually published in 1866, which of course in the United States was just after the end of the Civil War. One take-home lesson from this is when you're looking at genetic experiments, which is basically what Mendel did, you should be thinking about experiments that are going to be set up to do crosses between individuals where their genotype may be known from their phenotype, and the data are going to be recorded in lots and lots of numbers, and ratios are going to be calculated from them. Let's move forward a few years now and discuss Fritz Miescher. Miescher is from Switzerland. Miescher was from Switzerland. And one of the interesting things about Fritz is he was a bit of a black sheep in his family, because he grew up in a family full of individuals who wanted to, who were physicians, and it was assumed that he would do the same thing. Although he had different plans, he actually went to university and decided he wanted to be a priest. And that didn't go over very well with his family, by the way. And so he sort of conceded to instead that he would do some research, because he wasn't very interested in being a physician. And it's quite lucky for us that he decided to do this. So Miescher's work was really the foundation of molecular biology. A lot of times you'll hear people talk about Watson and Crick being the beginning of that, because they discovered the structure of DNA. But it was actually Fritz who was responsible for this. He's the discoverer of DNA, although he did not call it that. What he identified at first was just an unknown substance. He thought it might be a fifth protein. He wasn't sure about it. And one of the interesting things about Fritz is where he worked. And this figure on the right-hand side of your screen is actually showing the lab that he worked in. It was in a castle. And he was very excited about working here because he was working alongside some other German scientists, and he was very excited to be there. His lab was the old kitchen of the castle. It wasn't heated, and most of the time he was working at near freezing temperatures, and this was thought to contribute to his death. He died of pleurisy, of a lung infection, but some years later. And also, there was a war going on while he was doing this work. Anyway, he was basically a biochemist, and he isolated this fifth substance, this substance he thought was a fifth protein. And one of the biggest challenges that he had initially was trying to get enough material to work with. But Fritz was a pretty smart guy. He was working with some other smart scientists, one of whom suggested that he start working with a type of blood cell called a leukocyte, which was thought to be rich in this fifth substance. And because there was a war going on, Fritz had the idea of going to a nearby hospital and collecting the bandages. And basically, he did this to so he could collect the pus, because the pus would be full of these leukocytes. So whenever you're thinking that you got it hard, remember that you could be working in a freezing cold castle in the middle of the war and going to pick dirty bandages up from a hospital to get your work done. And just sort of continuing this line of sort of disgusting or difficult conditions that Fritz had to work in, when he was finally able to isolate this substance, this fifth substance, he wanted to determine whether it was a protein or not. And he'd heard about this substance called pepsin that some of his colleagues were using. We know now that pepsin is an enzyme. It's a digestive enzyme. It's in your stomach. And pepsin digests protein, so it's what's called a protease. So Fritz knew about pepsin and had an idea that he could take some pepsin and digest this fifth substance of his, this nuclein. And if he could actually inactivate it using pepsin, then he'd know he was working with a protein. Well, here's the gross part. These days, if you want to work with pepsin, all you have to do is go to a catalog, look up the number. A couple of days later, a package will show up on dry ice. Voila, you have some pepsin there to work with. Well, Fritz didn't have it that easy, and in order to find enough pepsin to work with, he had to go collect it from stomachs, and he actually did this by isolating pepsins from pigs' stomachs. So he went around to slaughterhouses, collected these stomachs, took them back to his freezing cold laboratory, isolated the pepsin from it, and then tried to see if that pepsin would digest his nuclein. It turned out it did not digest. And he also did some other treatments to see if it was a lipid or a carbohydrate or any other known substance at the time. And it turned out it wasn't. So this was a really big discovery. It was an entirely new class of biological molecules, and it would seem to be located in the nucleus, so he called it nuclein. This slide is showing a vial of nuclein that was actually isolated by Michener, and he isolated it from salmon sperm. He tried working with the bandages for a while, but even then was not able to get enough sort of high quality nuclein, and we know now that what he really got from those bandages was a crude nucleoprotein extract. So in order to get pure DNA, he had to go to a different source, and the source that he went to was actually salmon sperm. So this is a vial of nuclein, that's what he called it. We know it's DNA that was isolated from salmon sperm. And what's interesting about this, that salmon sperm is still the most inexpensive and readily available form of DNA. So if you're going to work with some DNA and you don't care what kind it is, that's what you're going to order from these biotech companies, salmon sperm DNA, thanks to Fritz Michener. And if we check back in on our timeline here to see what was happening socially around the time that Fritz was doing his work, we'll see that's right around the time, or was actually just before, when Alexander Graham Bell's telephone was patented. So you can imagine these scientists were working in these isolated conditions, and that communication between them was actually quite frustrated. At one point, Michener was quite interested in having his work on the nuclein be published, but it was actually received with some skepticism, because it's not every day that someone proposes a new class of biological molecules. And before he could get his work published, the scientist that he was working with in the castle wanted to verify all of these experiments. At this time, they weren't in the same location anymore. And so Fritz had to wait for this person to repeat all of his experiments, which he did, and confirmed Michener's conclusions. However, he had to wait for the war to get over before that would happen, so his publications were delayed for quite some time. And he was anxious about this and wanted to make sure that they very clearly wrote when he had first submitted the manuscript on the final publication. And if you look at publications today, you'll see that they're quite careful to say when the manuscript was first received and when it was first actually accepted. And the reason scientists do that is they don't want to get scooped. One of the really interesting things I want to point out right now is that even though that Mendel had done his work in 1865, which suggested something about hereditary material and genetics, this other work, the biochemical work that was going on, which we started with Michener and now we're going to look at Phoebus Levene, this biochemical work was actually being done in isolation. It wasn't until about 1930 that Mendel's work was rediscovered, and we started putting together the idea that this DNA molecule might actually be the one that's involved in heredity. So we're going to talk about Levene's experiments right now, keeping in mind that they really didn't have any idea of what this material actually did, but it was fascinating. It was in the nucleus. It was a new category of biological molecules. And Levene worked with another scientist whose last name was Jacobs, and they described the basic chemical composition of DNA. And they showed that it contained the sugar deoxyribose, and that also it had these 4 repeating subunits of adenine, thymine, cytosine, and guanine, and that they were held together by covalent bonds. What's interesting and really kind of sad about poor Phoebus Levene is that while in his time he was quite noted for his biochemical experiments, his legacy has unfortunately been one about the thing that he was wrong about, and that is this tetranucleotide structure that he proposed for DNA. He was starting to understand that there seemed to be some relationship between the amount of these particular bases that were present. He didn't really investigate it, and he proposed this structure you see here at the bottom of the screen. And what this is showing is that DNA structure is formed by phosphodiester bonds between 4 nucleotides, which are joined together in a circle. And of course, we know that the structure is one of a double helix. After Levene and Jacobs did their work, there were still a lot of questions to be answered about the structure and function of DNA. One of the first individuals to come along after Levine's work was Erwin Chargaff. And Chargaff was really not satisfied with this tetranucleotide structure. It didn't make sense to him. So he hydrolyzed DNA. And what does it mean to hydrolyze DNA? Think about a hydrolysis reaction. Okay, if you're thinking, you may remember, this is when a water molecule is used up, that's the hydro part, to split or lyse a macromolecule. So when he hydrolyzed the DNA, he split it up into its constituent nucleotides. And what Chargaff was interested in was the amount of each nucleotide that was present in a particular strand of DNA. This table shows you some of his results. On the left-hand column, you see the origin of the DNA. It ranged from human sperm all the way down to bacteria or E. coli. And then he calculated the percentage of each one of the bases in the DNA strands. And then he looked at the ratio of the different bases to one another. And what he found was that when he compared the amount of adenine to the amount of thymine, that the ratio was always very near to 1. And when he compared the ratio of guanine to cytosine, that the ratio was very near to 1. I want you to think about what his third column means, where he's comparing the ratio of AT pairs to GC pairs. And also think about what you might have concluded about the ratios of adenine to thymine and guanine to cytosine. Let's get back to our timeline now. Here is Erwin Chargaff, and he was really not too satisfied with this tetranucleotide structure. And he just thought there had to be more to DNA than just simple repetitive tetranucleotide blocks. So I encourage you to go and look at this slide. But for right now, I just want to point out that around the time that he was doing the work, this was around the time of the Korean War and when the first birth control pill was introduced, which was well before color TV. Not too many years after Chargaff was doing his work on the base composition of DNA, the work to really try to figure out both the structure and function of DNA continued in earnest. In the next section of Online Lecture 6, we'll talk more about the search for the structure of DNA by Watson and Crick. And we're just going to finish up this section by talking about some of the work that was done to try to determine the function of DNA. Now you'll remember from one of the earlier lectures that we talked about the Griffith and Avery experiment with the smooth and rough strains of pneumococcus. And that was one of the first experiments that was done to determine the function of DNA and suggested that DNA was the hereditary material. A similar set of experiments using different organisms was performed by Alfred Hershey and Martha Chase in 1952. Well, the work was published in 1952. And they used an entirely different system of viruses and bacteria. Their experimental design relied upon what's called a bacteriophage, and these are viruses that infect bacteria. The particular phage that they used was called T2, and these viruses have extremely simple structures. It's basically just a protein shell with a tail segment, and inside of it there is DNA. If you look in this next slide, you can see some of these phage structures here at the bottom of the screen, and they basically just have these tails that they attach to the bacteria with. They have a head that's shown here and then DNA on the inside. So bacteria were really, bacteriophages were really ideal for trying to determine the function of DNA and proteins, and specifically which of those was the hereditary material. The reason that bacteriophage were so good for this purpose is they have a very simple structure. It's just a protein coat and a nucleic acid on the inside. So what the Hershey-Chase experiments did was it took advantage of radioactivity. And radioactivity was important because it was a signal they could see later. So they could take a protein and label it with something radioactive that made the protein show up yellow. And they could also take DNA and label it with a different radioactive material that would make the DNA actually grow green. And so what they did was remember that these bacteriophage are viruses that infect bacteria. But before I go into the details of that, I want to just take a minute to point out that every time you hear someone talk about a simple or elegant experiment, it usually means they're working in a great system and had some good knowledge beforehand. And this was certainly true of Hershey and Chase. They'd actually been working on bacteriophages for some time, and so they already knew that a bacteriophage consisted of a protein coat and also that it had DNA at its core. They also had a very interesting piece of information, and that was from the Griffith and Avery experiments. They knew that someone had proposed that DNA was the hereditary material. So they realized then that they had the perfect system to try to test whether it was DNA or proteins that were in fact the hereditary material. They also knew that DNA contained phosphorus but no sulfur, whereas proteins contain sulfur but no phosphorus. So they were able to take radioactively labeled sulfur and synthesize proteins with it. So their proteins now had a label, a radioactive label that basically was like holding up a sign. Here I am, I'm a protein, I have radioactive sulfur in me. And they did the same thing with phosphorus. They had DNA that was synthesized with radioactively labeled phosphorus. So they started their experiment with protein and DNA that had been incorporated into bacteriophages but had been uniquely labeled so they could identify them later on. At the beginning of this experiment, they allowed the bacteriophage to infect the bacterium. And then on the top of your slide, you'll see their high-speed blender experiment that they did very quickly. As soon as the phages had actually attached onto the bacteriophage, they immediately took the solution and washed it, and basically, without disrupting the cell, knocked all of the proteins off the outside of the cell. And so they had a solution that just had cells in it and the protein code. And then they tested the liquid for radioactivity. And what they found was that the only radioactivity that was present in this particular preparation was radioactive sulfur. And that told them that the proteins actually did not make it into the cell. They did another set of experiments, which is shown on the bottom here, where they allowed this radioactive, they labeled the radioactive DNA, they allowed the bacteriophage to attach to the cell, and they allowed the material to be injected into the cell. And then in this case, they actually did an experiment where they took away the liquid. So they centrifuged all the cells down to the bottom and they washed them over and over again and took away the liquid. This would have been before I go into the details of that, I want to just take a minute to point out that every time all they had left was a simple or elegant experiment. And then they knew that means they were working in a great system and had some good knowledge those cells open. And this was certainly true of cell lysis, Hershey and Chase. When they tested, they'd actually been working on bacteriophages for some radioactive phosphorus. And so they already knew that from this set of experiments, Hershey and Chase included them, and also that it had DNA injected into the cell. They also had a very interestingly enough, they were actually very cautious about their results, and so it was only when they knew that someone had a number of others that DNA was together that it was fully accepted. So they realized then that they had a perfect system. However, this Hershey Chase experiment, whether it was DNA or proteins, forms the basis for this hereditary material. They also knew this concludes that part DNA 1 of online lecture 6. Phosphorus. Be sure to watch parts 2 and 3 before coming to class. Proteins contain sulfur but no phosphorus. So they were able to take radioactively labeled sulfur and synthesize proteins with it. So their proteins now had a label, a radioactive label that basically was like holding up a sign. Here I am, I'm a protein, I have radioactive sulfur in me. And they did the same thing with phosphorus. They had DNA that was synthesized with radioactively labeled phosphorus. So they started their experiment with protein and DNA that had been incorporated into bacteriophages but had been uniquely labeled so they could identify them later on. At the beginning of this experiment, they allowed the bacteriophage to infect the bacterium. And then on the top of your slide, you'll see their high-speed blender experiment that they did very quickly. As soon as the phages had actually attached onto the bacteriophage, they immediately took the solution and washed it, and basically, without disrupting the cell, knocked all of the proteins off the outside of the cell. And so they had a solution that just had cells in it and the protein code. And then they tested the liquid for radioactivity. And what they found was that the only radioactivity that was present in this particular preparation was radioactive sulfur. And that told them that the proteins actually did not make it into the cell. They did another set of experiments, which is shown on the bottom here, where they allowed this radioactive, they labeled the radioactive DNA, they allowed the bacteriophage to attach to the cell, and they allowed the material to be injected into the cell. And then in this case, they actually did an experiment where they took away the liquid. So they centrifuged all the cells down to the bottom and they washed them over and over again and took away the liquid. This would have been the liquid that they'd measured in the previous experiment. They took away all that liquid, so all they had left was a pellet at the bottom. And then they knew that pellet consisted of cells. They burst those cells open using a procedure called cell lysis. And when they tested that pellet for radioactivity, it was full of radioactive phosphorus. And so from this set of experiments, Hershey and Chase concluded then that it was DNA that was being injected into the cell. And interestingly enough, they were actually very cautious about their results. And so it was only when theirs and Griffith and Avery and a number of other experiments came together that it was fully accepted that DNA was the hereditary material. However, this Hershey-Chase experiment forms the basis for this on its own. This concludes part 1 of online lecture 6. Be sure to watch parts 2 and 3 before coming to class on October 23rd.